Well, hello everyone. Sorry for the technical difficulty. We're still trying to get our panelist, Dr. Margenot, connected, but we want to welcome everyone to the August Soybean Innovation Lab webinar, Principles of Soil Science for Managing Soybean Production, the Sub-Saharan African Context. My name is Courtney Tamimi, and I am the Associate Director for the Soybean Innovation Lab, and I will be your moderator for the webinar today. Today's webinar is sponsored by the Soybean Innovation Lab, a USAID Feed the Future initiative focused on improving soybean production and utilization in Sub-Saharan Africa. Today we are joined by registrants from over 30 countries, representing organizations within the private sector, academia, development agencies, and research institutions. So before we begin, I'd like to give everyone a quick overview of the GoToWebinar software, which you can use to engage with other panelists, ask questions, and access webinar resources. So whether you're using the desktop or web application to join the webinar today, you'll see a questions pane on your control panel. So please type in your questions as they arise, and we will collect all of your questions and address as many as possible during the Q&A session at the end of the webinar. Any questions that we aren't able to answer during the webinar today will receive a written response after the webinar has concluded. Also on your GoToWebinar control panel, you'll see a handouts pane. Access this pane to access today's presentation materials, the slides, the webinar agenda, and other useful resources. I'd like to give a quick introduction of today's panelists before beginning the webinar. Dr. Andrew Margenot is going to be taking all the presentations today. He is the lead soil scientist for the Soybean Innovation Lab. Dr. Margenot is an assistant professor at the University of Illinois, focused on soil fertility, water quality, and environmental systems research. Dr. Margenot has done extensive soils research in Kenya in collaboration with SIAT and brings a wealth of expertise to the Soybean Innovation Lab's programming across Sub-Saharan Africa. Also participating in the webinar today is Dr. Michele da Fonseca Santos. She's here on the video. Michele serves as the program manager for SIL's Pan-African Soybean Variety Trial Program, which introduces commercial varieties from across Sub-Saharan Africa, the US, Australia, and Latin America to fast-track the identification, registration, and release of new high-yielding soybean varieties. And you already know me, I am the Associate Director for the SIL program and manage the lab's day-to-day activities. And I will also be serving as your moderator for today's webinar. So to get started with today's webinar, we'd like to launch our first poll. Here you'll see it on your screen. We use these interactive polls as a way to get your feedback and to get an understanding of the range of attendees here with us today. So for our first poll, we'd like to know, do you have access to information on soil type in your area and soil testing facilities? And just select one answer for this poll. And so we'll go ahead and give everyone a few moments to record their answer. And you can go ahead and make your selection here on the screen in front of you. Just give everybody a couple minutes to record their responses. Okay, it looks like we've got most responses in. So we're going to close the poll in 3, 2, 1. Go ahead and close the poll now. And let's go ahead and share those results. So it looks like the vast majority of you, 57%, have access to both information on soil type in your area and soil testing facilities. And the rest of you either have access to neither of those resources or just one. So thanks so much for your feedback. This is really interesting feedback for SIL as we work to expand access to soil information and analysis for soybean practitioners.
Thank you, Courtney. Hi, everyone. Thanks for being here. I want to first apologize for that snag. We did a trial test of audio and video yesterday and this morning, and then, of course, 5 minutes out, it stopped working. So I apologize for that delay. So I want to today talk a little bit about the importance of soils for soybean production. The purpose of this webinar is to provide a pretty broad overview of how soils matter for soybean. The motivating question here is, why do soils matter and how do they matter for the for adoption of soybean technologies. So the term technologies is not just talking about the soybean plant itself and yield, but also other components of these bundles that SIL talks about. So things like lime, phosphorus, inoculum. It's not just the plant, but also the ROI on these inputs like lime that can be impacted by soil types. And as we'll see, there's some soil conditions that merit the use or the lack of use of some of these inputs like say lime or phosphorus. The goal for today is to go over three main topics. First is the principles of soils, some basic 101s on things that you should probably know to understand components 2 and 3, which are basically practices of management that can influence soils and as a result soybean. And finally, what are some of the takeaways for thinking about how to manage soybean via soils? Now, the overall goal of this webinar is for you to come away understanding some of some of the fundamentals of soil science as it relates to the adoption of soybean. I'd like to say that if you've taken moderate level soil science, this webinar might seem a little bit basic to you, and that's okay. What we're trying to do with this webinar is to get everybody on the same page. And furthermore, we would appreciate input on the exit survey because based upon your feedback, we will be designing future webinars on soils specific to the questions that you would like to see answered or discussion points. Next slide, please.
So we are going to launch, I think we already did this poll, Andrew, so I think we're good to go to keep moving forward.
Okay. Great, so let's start with why do soils matter for the successful adoption of soybean technologies? And here we're thinking about, again, not just the plant growth, the germplasm, but also these bundled technologies of inputs like lime, phosphorus, and inoculum. Next slide. Great. So I want to start by disseminating, or rather disabusing ourselves of some myths surrounding soils, especially in the continent of Africa. You may have heard the term tropical soil, and less so perhaps African soils. These are really egregious terms. I disagree strongly about them for a bunch of reasons. One is that they're not scientific. We have a system of classifying soils that reflects their properties, not so much their climate. And more problematically is that there are colonial origins to these terms that reflect a stereotype of the continent of Africa as being one kind of soil. And the problem with this is that there's in fact tremendous diversity of soils in Africa. So if one adopts a one-size-fits-all approach to soils in Africa, it's not going to work. The continent has arguably more soil diversity than the US or the EU. So we need to understand how soils differ in order to tailor and to target technologies like germplasm and inputs. So as a side note, what most people are really trying to get at when they say tropical soil are weathered soils, which are classified as oxasols, ultisols, ferrosols, such as the ones that you're seeing here on the right. These are from a few of our field sites, or rather SIAT's field sites in Western Kenya. These are common in the tropical belt, but that doesn't mean that all soils in the tropical belt are necessarily of this soil type, meaning weathered, acidic, classified as an oxasol. Next slide. So we need to really keep in mind that what I like to call soil context, the idea that a good starting point for thinking about adopting technologies like soybean is to ask, what soil type am I situated upon at your field site, at your farm? And a good place to start with this then is soil maps. Recently, or rather now 6 years ago, there was a very detailed high resolution soil map released for the continent of Africa, which you're seeing on the right-hand side. It has pretty high resolution, even at smaller scales. So it is, in fact, useful for practitioners to get a sense of what is the likely soil type that they're working with. This is helpful because there are some soil types that have what we're going to be calling static properties. These are unchanged. And there's implications for what soil type you're working with when it comes to things like, do I use lime? Do I use phosphorus? Or do I perhaps perform no-till versus normal till? Next slide, please.
Great, thanks so much, Andrew. So I think it's time to launch our second poll. Let's get that up for everybody to see. That was a great introduction. So everybody should see this poll on their screen. And Andrew, we're on the correct one. That's right?
Correct.
Okay, great. So right now we're asking what soil property is static and thus cannot be changed by management. So please select one of the options, pH, available phosphorus, structure, texture, or organic matter content. So it looks like we're getting some answers in. We'll give everybody just a few more seconds to record their answers. And as a reminder, be sure to type in your questions into the questions pane of the GoToWebinar control panel. And also ping me there if you have any trouble accessing the handouts, which are the presentation slides, the agenda, and a few more other resources. And I can walk you through how to find those. So Indra, I saw your comment and I'll respond to you to help you find those handouts. So we'll just give everybody a few more seconds. Looks like just about 2/3 of you have voted. If you haven't, please record your answer here. And we'll give everybody just 5 more seconds. 4, 3, 2, and 1. I'm going to close the poll now. And let me go ahead and share those results. So Dr. Margenot, you can see over half of the attendees got the correct answer of structure. Some other individuals thought, of texture. Some other individuals thought structure was correct. A few others pH. But Dr. Margenot, my understanding is that texture is the one soil property that is static and thus cannot be changed by management. Is that correct?
That is correct, yes.
Great. So everybody just keep that in mind. And all these poll results will be available to you after the webinar concludes. So let me go ahead and close this poll and let you have the platform again, Dr. Margenot.
Thank you, Courtney. Yeah, so lost the screen. So that quiz was to get a sense of what you all think about soil properties and which are more immutable and which are more pliable. So as Courtney has said, the correct answer was texture. I see that a lot of respondents chose structure, which is highly related to texture, but as we'll see in just a second, soil structure can be modulated by practices like tillage. And though there's a strong influence of texture on structure, one can move the needle quite a bit on structure depending on how you till or how you don't till. So the USDA Natural Resources and Conservation Service, NRCS, there's a link there at the bottom, thinks about soils, or rather proposes that we think about soils in terms of the properties that are static and those that are dynamic. So static properties are those that at human timescales over our lifetimes are effectively not changeable. In contrast, the properties that are dynamic can be changed. And so they typically are changed by practices like inputs and tillage. Now, this is important because static properties are simply what you have to deal with. We can't really change those. They might change over geological time, so over thousands of years. In contrast, we have an opportunity with dynamic properties to manage these to maximize soybean productivity. Next slide. Let's talk then about these very static and dynamic properties. I'm going to focus on a few of them. The first important one is texture, which, as we just saw from the poll, is a static property. So texture is the proportion of 3 particle-sized fractions known as sand, then the smaller fraction is silt, and then the smallest size fraction would be clay. And so it's the proportion of these 3 different particle sizes, sand, silt, and clay, that determine what we refer to as texture. So on the right-hand side, there's what we call the soil texture triangle. And this triangle encapsulates how the three different particle sizes, sand, silt, and clay, can come together to determine what we call the textural class. So if you hear people talking about clay or loam or a silty loam, they are getting at the relative proportions of these different particles. Now, as a side note, the word clay in soil science can mean different things, right? It is a particle size, less than 2 microns on the left-hand side, but clay is also a texture class, the clay texture class. And those are soils that have more than about 55% clay content. Next slide. Clay is something that we cannot change. It forms over geological time, and it strongly dictates how much organic matter can be held. However, there's still a lot of room for humans to alter the organic matter content of soils. So organic matter, also thought of as organic carbon, is a dynamic property of soils. Now, I think a good way to illustrate this is the photo that I took here of a farmer in Western Kenya. This is in Siaya County. And he had been practicing no tillage on the left-hand soil, and on our right hand, he had conventional tillage. And these are two different fields that are within 10 meters, and there's a strong difference in the color. The darker coloration on the left-hand side is due to higher carbon content. This is a nice example of how organic matter content is really quite pliable. Simply practicing no tillage for 5 years, as in this case in Kenya, can substantially alter organic matter. Now, we care about organic matter in soils because, well, it does a lot of things for soil fertility. Most importantly, perhaps, is that it acts as a reservoir for nutrients. It also improves soil structure. So let's talk about that a bit more. Next slide. So structure, as some people have sort of gotten at with their choice of this in the poll, is tightly coupled to texture. And it might seem that structure is a static property, but in fact, structure can be altered by humans. Structure can be thought of in a simple way as building a house with bricks and cement. And so the bricks would be the particles of sand, silt, and clay, i.e. texture. Now, what glues together those particles would be organic matter. So it's the interaction of organic matter with these particle sizes of the minerals that conglomerate or aggregate to make a structure. Now, there's inherent tendencies based on texture for a given structure. For example, a clay-rich soil will tend to have more of a massive structure or a blocky one, whereas a sandy soil might have a single grain structure. However, there's still a lot of room for humans through things like adding organic matter inputs by doing no tillage to change structure. Tillage, for example, will simply break up these aggregates, therefore it will decrease structure. Now, structure matters for soybean production because it tends to be a pretty strong dictator of water and how water is both captured, i.e. how it infiltrates, and then how that water can be held. It also influences soybean growth through things like compaction and the porosity of the soil. Next slide. Another property would be pH. So pH, as some people answered in the poll, tends to be strongly dictated by geology and by soil type. But that said, it can still be changed by humans. And so some of you perhaps have worked with liming to change pH. So obviously pH is in fact somewhat flexible. Now, we care about pH because pH, as some people think of it, is a master variable. It's the master switch. And if you toggle that switch, a lot of other things change. And perhaps the responses that we most care about would be the availability of nutrients. So think of this as pH controls how much you can take out of the bank account when it comes to nutrient elements like nitrogen, but really especially phosphorus and other metal nutrients like copper, zinc, boron, iron. And if you look at this diagram, it shows the availability of different nutrients as a function of pH, which is at the bottom axis. And in general, most nutrients, especially the ones that soybean needs the most of, NPKs, are of greatest availability in what we call the circumneutral pH range, from around 6.5 to about 7. So in general, we want to manage soils. We want to change this dynamic property of pH to achieve a pH of around 6.5. Now, if we look at the case of Africa on the right-hand side, looking at the pH map, we find that pH tends to track with precipitation. So the wetter parts of the continent have acidic pH. The drier parts tend to have a higher or an alkaline pH. Next slide.
Great. Thank you very much, Dr. Margenot. We're going to launch our next poll, but before I do that, I just want to note that we're getting some great questions coming in from you all attendees. So please keep your questions coming in. You can type them into your questions pane of your GoToWebinar control panel, and I'm recording those to share with Dr. Margenot when we get to the Q&A session. And as another reminder, we are recording this webinar. We will send a copy of the recording out to everyone after the webinar concludes, along with the presentation materials and the slides from today. So I'll go ahead and launch this next poll. This poll is asking about what aspect of soil in your area is of most interest and or concern to you for soybean production. So we're asking you to just select one that is of most interest and or concern to you for your soybean production. So we've got a few people already chiming in with their votes. Please take a look at your GoToWebinar screen and record your answer. We'll give everybody just a few minutes to, or a couple minutes to get their answers recorded. It looks like we've got about half of the attendees already voted. So get your votes in before we close. Okay, we'll give everybody just a few more seconds to record your answers. 80% of you have voted. Give a few more seconds. Again, what aspect of soil in your area is of the most interest and or concern to you for your soybean production? So you've got a few selections there. Please just select one, and I will close the poll in 3, 2, 1. Go ahead and close the poll. Let me share the results. So it looks like about 47% of you, so almost half, noted that nutrient deficiency is of most interest and or concern to you for your soybean production, followed by acidity and pH management. Lack of structure and poor infiltration took 3rd and 4th place, and then only about 8% of you are concerned with low organic matter content. So Dr. Margenot, I don't know if you have any comments on these poll results, but chime in if you feel like you do.
Yeah, I think this is quite useful for us at SIL to hear about, because I would have thought that acidity and pH might have matched the nutrient deficiency. Now, what's important to point out, as we'll talk about in the coming slides, is that nutrient deficiencies can be exacerbated or even caused by low pH. So a lot of times, by treating acidity, we can also alleviate nutrient deficiencies, especially for things like phosphorus and for metal ions like copper, zinc, etc. So this is quite useful.
Great, great. Okay, well, I'll go ahead and close these poll results now. And let's see, can we get, I just saw that you have a webcam available, so I thought maybe we'd see if you could flip that on, possible.
I'm looking at it right now, and it doesn't. Okay.
No worries, no worries. We'll keep rolling. Thanks everyone for your feedback. Really great response rates. We appreciate all the feedback, and again, getting some great questions in. So please feel free to type in your questions into the questions pane, and we'll get to those towards the end of the webinar. Go ahead, Dr. Margenot. I don't think we hear you. Let's see, we might have had a technical difficulty. Let's see if he's jumping back on. And oh, Everyone just bear with us for 1 minute while we get Dr. Margenot back online. 1 second. Oh, I think we've got Dr. Margenot back.
Yes.
Okay, great. The floor is yours.
All right. The Wi-Fi dropped there. Sorry about that. All right. So let's, given this background of soils and the basics, and also given that latest poll, let's dive into some more details about how practices of management can influence soils and as a result can indirectly or directly impact soybean productivity. Next slide. So before we talk about the practices, I think that we should start with the basic science overview of soil fertility. So there's really different components of fertility, and not unlike soil health, we can think about these as being a triumvirate of chemical, physical, and biological. And we tend to think of soil fertility in the first sense as being chemical. So here we're thinking of things such as the pool or the availability of nutrients like nitrogen, phosphorus, copper that are available in soils for plant uptake. Keep in mind that for all plants, except for carbon, every other element that the plant needs to grow is contained in the soil. There's also potential elements that can cause growth problems to soybean, such as aluminium in soils. And so there's also this potential toxicity component to the fertility, chemically speaking, of soils. Soil fertility can also be thought of as being physical in nature. So here, thinking about things like compaction and how well the soybean roots can penetrate and can grow. Indirectly, physical fertility deals with water. So when you have a rain-fed system, you want to be able to capture and to harvest that water into the soil. And so having good soil structure, for example, will permit infiltration rate and also good water holding capacity for a rain-fed soybean system. The final component of soil fertility that we manage is biological. And so we tend to think about microbes and soil health. For soybeans specifically, and especially for the context of Africa, there's a very important role of a symbiotic bacterium that is involved in end fixation in the root nodules of soybean. There's also some soils may carry pathogens, which we won't really talk about today. And finally, some soils have certain communities of microbes that can form associations with soybean roots that overall promote plant growth. So this is different from end fixation. Next slide. Let's talk about chemical fertility of soils. So when we think about the elements that are important for soybean growth, we said how carbon is fixed by the plant from the atmosphere. That's photosynthesis. Nitrogen is also kind of unique because here too, the soybean plant can take it up from the soil, but a large budget of the nitrogen that the soybean is using for its tissues can be provided also from the air via the symbiotic microbe in the root nodules of the soybean plant. But the other elements are all going to be acquired from the soil. So here we're thinking about phosphorus, potassium, calcium, and magnesium as the main macronutrients. Then we've got the micronutrients. And so these are nutrients, as the name implies, that are needed in lesser amounts, hence micro. For example, soybean leaves tend to have the macros as a percent of their mass from 2 to 0.2%. Whereas the micros, we think about those and we measure them in soybean biomass with the units of part per million or ppm because we need very small amounts. There's also some elements in soils that can be toxic to soybean. Aluminium is perhaps the most important in soybean, soybean is distinct in that it's especially sensitive to high levels of aluminium. As we'll see in just a second, that's also a result of low pH. And of course, there can be too much of a good thing. So there's also some nutrients like manganese or boron that at very high concentrations in soils can cause toxicity to legumes, including soybean. Now, soil pH is an important part of the chemical fertility, but it's not itself a nutrient. As we've discussed, though, it is a master switch that dictates how available this nutrient bank account is for the plant to take out from. Next slide. Let's talk a little bit more about the role of pH in modulating chemical fertility. So it's not just enough to have a certain total amount of nutrients, like say phosphorus. It's also important that they be in a form that's available for the soybean plant to tap into. Now, pH is important as well because it can affect the potential of the soil to store these nutrients, either the ones that are already present or that we're adding as fertilizers. So on the left hand, we have a diagram of cation exchange capacity, also known as CEC. And what we find is that at low pH on the left-hand side, most of that storage capacity of the soil is dominated by non-nutrient ions like aluminium. Again, that's toxic to roots in high concentrations. And as we increase pH, we tend to have a swapping out and getting rid of the aluminium and instead using that binding potential to store nutrients for soybean. So this will be why liming is very useful, not just to decrease AL toxicity, but also to increase the potential holding capacity of nutrients that you want there. A final note on the role of pH and metal elements. We know that MO, which is molybdenum, is really important for N fixation because it is a cofactor for the enzyme used by the bacterium in the root nodules to fix N. So we can induce a deficiency of this element sometimes as a function of pH. And if so, there can be what looks like a N deficiency. And so that might seem odd because we talk about how soybean can fix its own N via these root symbiotic bacteria. But if they don't have this metal ion, then they themselves won't be able to perform that function. So again, this is just to emphasize that pH has multiple and very complex impacts on soybean. It's probably a good place to start when thinking about how to manage soils for soybean productivity. Next slide. So then the final component of soil fertility that I'd like to touch upon is the biological one. And as we just mentioned, for soybean, the one that really comes to mind is the rhizobacteria, Brady rhizobium, and specifically the species of Haponicum, which is again a symbiotic, meaning that it needs to partner with the plant, in this case soybean, to survive. The soybean plant provides photosynthate, so sugar, and the bacterium, in this case, fixes nitrogen from the gaseous form into the plant available ammonium form. Now, most soils in the continent of Africa are thought to not have naturally occurring levels of this symbiont. And so this is why we inoculate seeds with this specific symbiont. Now, there's different emerging lines of evidence that there might be indigenous and fixing rhizobacterium that can perform this function for soybean. But long story short, the consensus seems to be that we need to inoculate to improve this aspect of biological fertility for soils specifically in Africa for soybean. Next slide. Now let's talk about practices that can influence these three components of soil fertility with implications for soybean yield. So the first one that I want to touch on is tillage. What you're looking at here are two good examples of tillage being practiced by smallholders. This is again some work being done with SIAT in Kenya. On the left, we have conventional tillage, and this is all being done by hand with the djembe, and that's pretty typical of smallholder systems, as you likely well know. And on the right-hand side, we've got also smallholder hand-based tillage, or in this case, no tillage, with residues maintained on the surface as a mulch. So these two components are two principal tenets of what's called conservation agriculture, keeping some kind of a surface cover and trying to minimize disturbance of tillage. So why would we want to minimize tillage in some cases for soybean? Next slide. So it seems like people tend to talk about no tillage and conservation ag as always being good. And that's not necessarily the case, depending on climate and depending on soil type. And there's a big debate about this for, quote, Africa, which is, again, so diverse in climate and soil types that it might be a little bit reckless to have one blanket recommendation of no tillage always working. So here's some data from the US that suggests that there's some contexts in which no tillage is beneficial for soybean, but in other contexts, depending on climate, it's detrimental. So what you're looking at is a recent study that assessed when does no tillage increase soybean yield and when does it not affect it or even hurt yield. And the consensus based upon this map on the right-hand side is that when you have drier climates that are more prone to low soil water content, i.e. drought stress, no tillage does pay off. And when you've got cooler and wetter climates where water isn't a big issue, no tillage can actually hurt yield because, well, the soil stays too wet. So this is a good example of how in warmer climates and in soils that have more clay that are harder to for roots to push through, no tillage can be of benefit. Next slide. Another major practice is liming. So now liming, as we've been getting at, is a way to manipulate soil pH and specifically to increase soil pH. If you are in alkaline soils, a pH of about 7.5 or higher, then you want to acidify, and lime is not what you want. That would be sulfur. But for, I think, most areas of production in Africa for soybean, we're looking at acidic soil. So we're going to focus on liming as a practice to manage pH. This is a photo from some of our sites in Western Kenya showing, I don't know if you can see this, but on the left-hand side, we have a farmer who's measuring pH on his unlimed control plot. On the right-hand side, we have the right next door limed plot, and the pH is 5.4 to 6.3. This is a nice example of how a small dose of lime, in this case 2 tons per hectare equivalent, can increase pH to move the needle into the optimum zone. Next slide. So just as pH has multiple complex controls on soil nutrient availability, well, so too lime, by changing pH, has multiple benefits in acidic soils for crop production. So the first is that liming can decrease the toxicity of aluminium to roots. And so this is kind of tough to untangle from a nutrient deficiency because AL basically stunts roots, and so these roots can't take up nutrients like, say, phosphorus. And that might look like a P deficiency when really it's AL toxicity. Philosophically, this gets kind of blurry, but the point is that liming can decrease toxicity in very acidic soils. We're talking less than 5.5 or so. The second benefit that co-occurs with lime is that as we move up pH towards 6.5, we free up nutrients that are already present, but in chemically locked up forms, in particular phosphorus and metals like copper and zinc. Third is that if soils are very, very acidic, it can be detrimental to N fixation. One reason for this is that molybdenum can be tied up potentially, slash acidic soils tend to be naturally deficient in MO. And so if we alleviate this constraint, then we can have more N fixation. And finally, we can increase the return on investment of additional inputs. So thinking here about the bundles that SIL promotes, where we have inoculum, lime, and phosphorus, lime can increase the return on investment of the phosphorus component of that bundle. And this is because as we go above pH 6, 6.5, the phosphorus that we're adding is more likely to be used by the plant. This is because at lower pHs, phosphorus gets bound up to metals and can't be used by the plant, and that would be a waste of that resource that we're adding. So to sum this up, we know that soybean is very sensitive to AL toxicity, as well as suffering from locked up nutrients at low pH. Liming alleviates this pH constraint specifically for soybean. It has more benefits. Next slide. The final practice that I'd like to touch upon is pretty obvious, right? It is fertilization. So fertilization is perhaps the easiest way to increase crop production, including soybean. As we've just discussed, it makes sense to have the other things in place, specifically manage the pH first, and then maximize the nutrients that you're adding as a fertilizer. So on the right-hand side, as an example of the benefits of adding something like, say, phosphorus, we have a plot showing the 1-to-1 ratio of a control plot versus adding P and or inoculum. These are from on-farm trials in Nigeria looking at soybean. And what we see is that most points are above that 1-to-1 line, meaning that adding phosphorus and or inoculum increases soybean production in smallholder plots in West Africa. So we typically don't think about nitrogen when it comes to soybean because it can fix its own end. It makes it, I think, a special crop because we have one less nutrient to think about, unlike, say, maize. That said, if there's high amounts of background nitrate, either from adding it as an input or just naturally, that can deter end fixation. So when we talk about soybean and inputs, we're really focusing on phosphorus and then potassium. The micronutrients are also important, especially in very weathered soils that are naturally deficient in these. And the good thing about micros is that you don't need a lot to make a big splash, hence the name micro. So it's easy to add small amounts on a mass basis to have big dividends in soybean growth. Next slide. So these are just examples of how soil fertility that's not well managed might manifest in visible symptoms of nutrient deficiency. So we have phosphorus. The purple veining is pretty typical of P. Then the other major nutrient that we should think about for soybean is potassium. And that manifests when it's deficient as this chlorosis on the outer edge of the leaves. Now, potassium is trickier because it's in part influenced by the water content. When things get dry, potassium is less available. And this goes back to soil structure and how having a good structure to capture that rainfall can help mitigate the risk of K deficiency. Next slide.
Great. Thank you very much, Dr. Margenot. Just really interesting, really informative. We really appreciate your insights. So I'm going to launch the next poll. This is the second to the last poll, so we really want your feedback. So we're curious, in your area, do you have access to the following inputs? And for this one, we'd like you to go ahead and select all that apply. So we've got lime, phosphorus, nitrogen, potassium, and organic matter, for instance, manure or compost. So we really want to know kind of what you have access to in your area in terms of inputs that can affect soil fertility and soil quality. So I'll just give everybody a few seconds to respond. And again, we're getting a lot of great questions coming in quick, so I'm trying to respond to everybody, and we will get to as many questions as we can during the Q&A portion of the webinar. Just as a reminder, we will provide written responses to any questions that we aren't able to address during the live webinar, so your feedback is being recorded and you will get a response. So about 80% of you have voted. Let's see if we can get a few more people giving their feedback about what inputs they have access to in your growing area. And again, you can select all that apply. So I'm going to close the poll in just a few seconds. In 3, 2, 1, I'm going to go ahead and close the poll. Let me share the results. So it looks like we've got some pretty widespread feedback, which is good. Looks like lime is the input that is least available or accessible in the areas for our attendees. Nitrogen is widely available, followed by organic matter and phosphorus. Potassium a little less so. But we do see that lime, which is something that we've noted in some of the Soybean Innovation Lab research that we've conducted, is a little less available. So Dr. Margenot, I'll let you chime in if you have any comments on these poll results, and then we can keep moving.
Thank you, Courtney. This is also a really good poll to get answers to, because I think this shows well that people voted how acidity was the second most important consideration, or what they perceived to be the biggest soil-based constraint. They had voted for nutrient deficiencies to be number one, but lime seems to be the least accessible. So this is interesting because it suggests that increasing access to lime might be a good way forward. The availability of nitrogen is important, but as I've mentioned, for soybean, it may not be as important, right? Because soybean can provide a lot of its own nitrogen under the right conditions. It is good to see that phosphorus and potassium are available almost at the level that N is as well. So,
great, thank you for the feedback. And sure. Just everybody, we had a little late start with the AV, so if you can just hang on a little bit past the hour, we will still have a Q&A session. It'll just be a little bit delayed, but we will stay on the line, so hopefully you all are able to as well. So Dr. Margenot, I'll pass it on over to you.
Great. So let's talk about some of the takeaways of all of this knowledge. We've talked about basic soil types. We've talked about the science and the chemistry of fertility, as well as the biology and the physical aspects of soils when it comes to soybean. There's some pretty obvious implications here then for how one acts on these. So the first thing that I'd like to propose is that we should think a lot about starting with a soil test. And this might seem obvious, but you would be surprised. Even here in the Midwest, not everybody tests right away. And this makes sense. Tests are expensive. Having worked in Kenya quite a bit, I know that it can be difficult for some farmers to have access to a lab, or if they do, it's maybe not that affordable. But I'd like to propose that testing is really important because it can let you allocate resources more efficiently. For example, if you test your soil and you've got a high amount of phosphorus, there's no need to spend money on phosphorus. So, and when we think about testing, there's different ways to break this down. We want to think about tests as a way to initially determine what we need to do. Then second, correct what is needed to be done. And finally, soil testing thereafter can be used as a check-in. So this gets at the central concept in agronomy of build up and maintain for soils. The idea of build up and maintain is that you want to increase soil nutrient levels to a threshold that are adequate, not too much, but not too little. And then you want to maintain that. And the maintenance comes about by doing some calculations on how much nutrient is being exported with the grain harvest. You want to replenish that amount, those kilos per hectare of PK down the line, with what is being taken off as soybean grain itself. So to make this more concrete, let's go to the next slide. And let's realize that when we harvest any crop, be it maize or millet or soybean, the grain that we're harvesting, or even the fodder for feeding, say, cattle, that contains nutrients that came from the soil, as we talked about. And so if we're not replacing those, then ultimately we will be mining the soil of its nutrients. And over the course of, well, it might be as soon as 2 years, as long as 20 years, but eventually we will run out of that nutrient. Again, N is the exception for soybean because it can fix its own N. What you're looking at here is an example of nutrient partitioning of soybean. So on the x-axis, we have the growth stage, the different stages of soybean development and growth. And on the two y-axes, we have the amount of nutrient uptake being shown here for not just the dry weight, but also for phosphorus on the bottom left, and then for N on the top right, and then for potassium on the bottom right. And there's two ways of looking at the uptake and the distribution of NPK across different biomass fractions. On the left-hand y-axis, we have the amount per hectare. So for example, for phosphorus, we see that by the very end at harvest, there's an equivalent of about total 20 kilo per hectare of P2O5 that the soybean has taken up. That's how much P came out of the soil into the biomass of that soybean. And then the other way of viewing this data is on the right-hand y-axis, which is the percent of total of that nutrient. So going back to phosphorus, that would be, if you look at the blue color, that would be indicative of grain. The legend is the top left here. So that means that that blue swath of phosphorus is about 80%, meaning that 80% of all of the phosphorus that soybean took up as a plant ends up in the grain. So when we harvest that grain, the 20 kilo per hectare of phosphorus, 80% of that is in the grain. That's about, my math is fuzzy at this time in the morning, but it's about 16 or so kilo per hectare equivalent of phosphorus that we're removing from the soil of that field. And so to sustain or to maintain a available pool of phosphorus for the next year's crop, we want to fertilize at that rate. Next slide. So this is what build up and maintain means, is that we want to build up to a soil test level that is appropriate, that's adequate for soybean. And then we want to maintain that level by replenishing what we're exporting with a new input. Now, this is a way to be very efficient with our resources. So we don't want to over apply phosphorus. We first do an initial soil test to figure out how much should we be adding to build, if we even need to add any. And then we do a periodical soil test to check if our calculations are correct. And year to year, there's not necessarily a need to do a soil test as long as we're doing calculations on, well, if we have this many kilos of grain that we're harvesting, how much P do we need to add back to replenish what was taken off? Next slide. The other major recommendation that would result from an initial soil test would be if it's an acidic soil and if it's below 5.5, then there's a critical need to add lime to increase pH. Now this can be done in two ways. One is by broadcasting, but you'll find though is that for very acidic soils, especially for weathered soils, we need a lot of lime. And think here of Brazil, they've got the same soil types as much of East Africa, for example. They need to add up to 10 tons per hectare of lime. And that's quite a big investment, especially for smallholders. So it's sometimes okay, or sometimes it is equally effective to what's called spot apply, also known as the bottle cap method, where you can, in a small bottle cap, add into the planting hole where the seed goes a little amount of lime. And that's what's part of the SIL bundle. The whole point here, though, is that if you get a soil test pH measurement back, and if it's less than 5.5, that's thought to be the critical zone. People talk about 5.8 to 6 as the optimal pH for soybean. In any case, getting above to around 6, 6.5 is optimal for soybean productivity. Next slide. So to summarize, we began talking about how soils are oftentimes stereotyped for the African continent, when in fact they're more diverse in the continent than all of the US and the EU combined, arguably. And as a result, we have to be cognizant and aware of this diversity simply because it makes sense to get a better, efficient use of inputs. We want to target how we manage soybean depending on soil context. Now, soils have some properties that are static, and here's where soil maps, soil type, is really important to know your texture. It also can give you clues to the tendency of your soil, even for the dynamic properties. So for example, structure, pH, or dynamic, we can change it, but they will be strongly influenced when you start depending on soil type. So there's still value of soil maps for determining how to manage the dynamic properties. Finally, the properties that we can manage that are dynamic have indirect and direct effects on soybean via soils. And specifically, we've talked about physical, biological, and chemical fertility, and we can influence these by tillage, adding lime, and adding inputs of nutrients. Soil testing here is critical, both to identify if there's a need and for what nutrients specifically, as well as how much of these things to add, whether it be phosphorus and or lime. So with that, I think we're ready for questions.
Great, thank you, Dr. Margenot. Do you want to launch this last poll? Is that of interest?
I think that'd be great.
Okay, so let me go ahead and launch this last poll and then we'll get to questions. So this poll is asking, in your area, what is the relative importance of soil fertility management for soybean production compared to other factors? So we want to know, how does soil fertility management rank in terms of importance when you compare it to many other factors that you as producers or private sector partners or NGOs might be facing? Is it very important, equally important, somewhat, not really, or not at all? So if you can just give your feedback, and then we will move right on to questions. I'll give everybody. Great, we are getting quite a few feedback in. Looks like about 75% of you have voted. I'll just leave this open for another couple seconds. And I will go ahead and close the poll in 3, 2, 1. Closing the poll. Let me go ahead and share the results. So it looks like over half of you note that soil fertility management is pretty important. It's equally limiting as other factors for your soybean production. And then you're pretty much split between it being very important and somewhat important. So nobody's saying that it's not important at all or not very important. So that just shows the importance, and I think the importance essentially of this webinar to the challenges you're all facing. So Dr. Margenot, if it's okay, we can move right into the question and answer session.
Sounds great.
Okay, so we are now into the Q&A session. I've got a lot of great questions for Dr. Margenot. Again, if you can hang on the line, we will get to as many as we can for the next 10 or so minutes. And if you do need to leave, please do complete the short exit poll that will pop up when you leave the webinar. This really helps us with feedback about how the webinar was, any suggestions you have to improve the webinar, and of course you can always email us at soybeaninnovationlab@illinois.edu with your comments. So let's go ahead and get started. So Dr. Margenot, one question came from James Njoroge, who is with the Kenyan Agricultural and Livestock Research Organization. And I will ask you, Dr. Margenot, if you can keep your answers just as brief as possible so we can get to as many questions as we can. But James asks, apart from adding lime, what other practices can the farmer carry out to improve soil pH?
That's a good question. Apart from adding lime, well, it would be any source of calcium and or magnesium to increase pH. The lime works in two ways. You've got the calcium, but also the carbonate. And so if you have either of those in a non-lime form, there can still be some benefit. A good example here would be gypsum, which is calcium sulfate. It improves soil structure, but also the calcium can help displace the Al from the soil clay surface, and that then can be washed out. That said, though, there's really no alternative to using lime. In the end, you want to use something that has calcium carbonate. One possible alternative could actually be biochar or some kind of ash. Ash basically is rich in calcium oxide. That's a liming agent. The trick there is that there needs to be quite a bit of biomass burned to provide enough ash to match a liming dose.
Great, thanks so much. Emmanuel Sangodele with OLAM International asked, what is the critical limit for macro and micronutrients for soybean production in Africa? So that might be a little tough to nail down, but he is asking, what is the critical limit for macro and micronutrients for soybean production, if that's possible to answer?
Sure. So that's a pretty broad question, and so the answer would be, we'll probably be here for 2 hours. I'll say this, it's the favorite answer of any soil scientist. It depends. It depends on your soil context, basically. It might depend also on the variety of soybean. In general, though, we can talk about specific thresholds. And so for this, I'd like to refer you to the handout that has, I believe, a table on thresholds recommended for soybean. And these are thresholds based on the MELIC 3 extraction for phosphorus and for potassium. So for phosphorus, I'm going to pull it up right here. Typically, what we think of as an adequate threshold for the MELIC test would be around 26 to 50 part per million. And for potassium, it's about 60 to 180 ppm. So if you're beneath 25 for phosphorus and beneath 60 for potassium for MELIC extraction, those are thought to be conditions where there's a yield response to soybean. Now, a lot of these data are being used from US sites. So again, this goes back to context. Knowing what soil type you're on, where that can be useful to match your soil type in Africa with soil types that are similar in other places that have done site-specific trials on these thresholds.
Great, thanks so much. We're just pulling up the handout that Dr. Margenot referenced. You should see it on your screen now. This is available in the handout section of your GoToWebinar control panel. And so there's some good content in there for you all. We have a few more questions. Yurga Masrashah with the Ethiopian Institute of Ag Research asked, can nutrient deficiency aggravate diseases and pests? And how can we differentiate the symptoms of a disease from a nutrient deficiency?
That is a great question. And as for the latter, that can be tricky. A good example is my lab works a lot with coffee production, and coffee leaf rust looks just like potassium deficiency. And it really is difficult. Visually, when it comes to soybean, like most crops, one has to be an expert. I have trouble with this. The ultimate solution is really to do a foliar test and or a soil test to determine whether it is a pathogen or whether it's a nutrient deficiency. Courtney, could you, I forgot the first part of that question. Do you mind refreshing my memory?
Sure. The first part of that question asked, can nutrient deficiencies aggravate diseases and pests?
Thank you. Yeah, so I think the short answer is yes. And a good metaphor here would be human health. If we're deficient in calories and protein, that can aggravate what might be minor things like a flu. So too with crops, there can be more susceptibility to pathogens if there's deficiencies in nutrients.
Great, thanks so much. The next question comes from Haroon Maruthi. Haroon is a SIL collaborator based in Kenya as part of the ORISE program. So Haroon asked, how often should a farmer conduct soil testing? And a second question related to how often should a farmer apply inoculant, every season or after 2 seasons?
Okay, I'll start with the first question. So how often should a farmer soil test? Well, as we discussed in those last few slides, I think starting before the planting of a new field, that's a good time to take a soil test. And then after that, every, I would say 2 to 4 years, if a farmer is doing the nutrient balance. So by that I mean is that you take the first year soil test and you know what you're starting with. And then you want to be fertilizing with what you're exporting based upon soybean grain yield. You know the P content, the K content of that grain, and you can then do some math and, well, how many kilos do I have to add back to make up for what we're exporting? And so if you're doing that kind of math, think of the soil test as a way to check that the math is correct. So in this case, even every 4 years is a typical practice for a lot of soybean farmers in Brazil and the US. The second question I've also forgotten. So Courtney, do you mind reminding me of that?
No problem. So Haroon also asked, how often should a farmer apply inoculant, every season or after 2 seasons?
Gotcha, thank you. This I think would depend on whether, I do not know, that's the short answer. I think this would depend on the site. From what I understand, it might take repeated use of inoculum to establish a indigenous community in that field. And so this might take years, it might take decades, it kind of depends. I don't want to misspeak because I'm not an expert on inocula. But I will say that it's probably best to keep at it for many, many years, maybe the first 5 years, in a soil in a continent like Africa, where this bacterium is not indigenous. It's not native to the continent. It came from Asia. And since soybean has a relatively recent history, there may not have been enough time for this to be established. So in the case of the US, there's also this issue that Africa now has with soybean. When farmers here began to grow soybean, there were no native rhizobacterium for soybean because, again, it is a crop from Asia. So what the US did in the 1800s was actually to ship soil from Japan, and they mixed it in with soils in US farmland. Today, this would never happen because of concerns about importing pathogens. And in fact, people think that soybean pathogens got imported from these Japanese soils. So this probably won't work for Africa today, just because of the amount of soil that you would need to bring over from Asia. But the point stands is that if you inoculate repeatedly, you can eventually establish enough of a colony in the field that you should be able to, down the line, not have to inoculate. The takeaway from this is that I think it might be good after several years to do a small test plot and to not inoculate in that part for those seeds and see what their responses are.
Great, thank you. A lot of people are still hanging on the line, so we're going to get to a few more questions yet. Mboa Arnold with AATF in Rwanda noted that in Uganda they have lots of ash from cooking places, and can this replace lime?
Definitely. In fact, that is a phenomenal source of lime. There's different kinds of lime. So we say lime, we think of calcium carbonate. There's also dolomitic lime, which has magnesium carbonate, but there's also things like quicklime that you may have heard about, which is some kind of an oxide or hydroxide. So they're not carbonates, but they're still being oxides or hydroxides. They still strip away protons, or they consume protons, which is in part what drives low pH. And so ash, be it from slash and burn, be it being from fires, either for cooking, ash contains a lot of calcium and magnesium oxides and hydroxides. And so it's a very good source of quicklime, or basically it is a good liming agent. So I would say, yes, that's a very good source. Also think about, by virtue of combustion, you're also concentrating lots of nutrients like phosphorus and some metals like copper. So ash has the additional benefit of being a source of nutrients, not just lime.
Great, wonderful. Let's get to maybe one more question, maybe two. I don't want to keep people too long past the hour, but Matiku Haile Mariam, sorry if I butchered the name there, with Mekelle University in Uganda, had a couple questions. First, how would you stabilize aggregates? And then second, he noted that some farmers want to rotate soybean with sesame grown on clay soil. And in your opinion, is this a good idea?
Sure. So how does one preserve these aggregates? I would say that you can only not just preserve, you can even build and increase and augment the amount of aggregation. In general, we want to preserve, or rather one can preserve aggregates through reducing tillage. Tillage is the best way to break up these aggregates. That's what tillage is, right? And the other way would be to add a good amount of organic matter. This is where conservation ag can be quite helpful, because the two central components of conservation ag are to reduce tillage and then to keep some kind of a surface cover, as well as roots in the ground. And so the last two, keeping roots in the ground like a cover crop or constant cropping, and also keeping a mulch, those are effectively sources of organic matter addition, because aggregates are the result of the organic matter glue of the particles of sand, silt, and clay. Combining manure residues with less tillage is the best way to promote aggregation. The other question was about following soybean with sesame in heavy soils, as they're termed. Also, sounds like they're clay rich. I don't know if these are oxasols or if they're vertisols. Vertisols are also called black cotton soils. You find them in the valley bottoms. In either case, they're very clay heavy, or rather high in clay, called heavy. Tillage is almost necessary to work these soils, especially if they're surface crusting. We found in some trials in Western Kenya that conservation ag can decrease yields, because if you don't till, then the surface of the soil hardens and all the rainwater just runs right off. So again, this is why.
And Dr. Margenot, Matiku chimed in to say that they are vertisols.
Vertisols, okay. Yeah, so with vertisols, no tillage might be tough because you do need some mechanical breaking up to allow water capture in some cases. I don't think there's any reason to suppose that following soybean with sesame would be detrimental to either crop. So I would say go for it if that's a suitable cropping choice for your region.
Great. And I want to make one correction. I mistakenly said that Mitiku was from Uganda, but he is from Ethiopia. I should have noticed from the name, but sorry about that, Mitiku. Let's get to one final question. We've still got quite a few people hanging on, so I think the questions and answers are really valuable. Dick Tinsley with Colorado State University in the U.S. asked, what adjustments in soybean management do you suggest when working with oxysoils or Andy soils?
What adjustments in
soybean management do you suggest?
And it's oxysols and andisols. Well, the common shared component of those two soil types would be a natural tendency towards phosphorus fixation, and they act in different means. So in oxysols, the natural status quo is you've got pretty low pH, and you also have an abundance of mineral types, mostly aluminium and iron oxides that can fix phosphorus. So in this case, by the way, phosphorus fixation is not the same sense of the word of fixation as nitrogen fixation. So N fixation is a good thing. It's a way that we can increase N in the soil. P fixation has the opposite meaning, confusingly, which means that we bind phosphorus to iron or to aluminium, and in doing so, we lock it up. So P fixation happens when you have low pH and you have that mineralogy that tends to be common in weathered soils like oxysols. So in the case of an oxysol, the solution will be, again, context specific. It is to lime. So here, the Brazilians, I think, are a shining example of how you can use lime to deal with oxysols and get great soybean productivity. They're, in fact, one of the most productive soybean producing regions in the world, and they have oxysols. So liming for the oxysol is a way to decrease P fixation. In the case of the andisol, we have a very different soil type. We have not that much clay. We have clay-sized particles, but they're not heavy, so to speak. Andisols, though, also have severe P fixation if they're of the glaseous or the vitric andisols. And these andisols, like what you find in Japan, will also fix phosphorus, but they do so with a different kind of mineral. And here, liming won't help you. There's no real good solution here except just to throw on the phosphorus. And the Japanese are experts in how to deal with phosphorus and andisols. One approach that they've been using, besides just dumping on a whole lot of P, which is costly, is to coat the seed in phosphorus. So for example, with potatoes, Japanese farmers tend to add phosphorus right next to the tuber as a way to minimize the chance of P fixation. So a theme here could be the placement of the phosphorus, not broadcasting it as a granule, but adding it as a spot application or as a furrow application. Just like we talked about adding a little bit of lime right next to your soybean plant, so too in a planting hole, adding the phosphorus in an andisol would be effective. Besides that, there's no real reason to think that oxysols or andisols are any different. Once you deal with the issues of pH and P fixation in the oxysol and P fixation in the andisol.
Great, thank you very much, Andrew. So I think we probably need to wrap up here. There's a few questions that we weren't able to get to, but please note we will share these with Dr. Margenot. He will provide, with our help, a written response to you all. Thank you all so much for your participation today. Thank you for your patience with the audio video issues we had. Thank you so much to Dr. Margenot for all the insightful responses. And again, thank you to our wonderful attendees. So this concludes the Principles of Soil Science for Managing Soybean Production, the Sub-Saharan African Context webinar. Thank you so much again for your participation. Today's webinar has been recorded, and a link to the recording will be sent to you in a follow-up email. When you exit the webinar today, please complete the short exit survey. We greatly appreciate all your feedback and the time you spent with us today. So please, if you can, take a few moments to complete this exit survey. We do rely heavily on your feedback to learn how to improve our webinars and find out what content you're interested in for future webinars. So thanks so much again for your participation today, and have a great rest of your day or evening. Thanks so much. Goodbye.